had roughly 140 plus caucuses so far in the last 15 years. And today I think we'll have a very exciting and interesting one. You know, I want you to know that the Joint Steering Committee for Public Policy that puts on these caucuses, you would welcome to any one of the offices of the co-chairs any of the recommendations and suggestions on particular topics, heart disease, cancer, we see obesity around all the time now. Any particular topic you might have, you might believe of interest. We would really enjoy it if you would bring it to our attention. We have, of course, the four co-chairs of the caucus, Sherry Porter, Lois Katz, Rush Holt, and now Mike Castle from Delaware. These are people who, in the last several years, particularly in those 5 glorious years between the close of the year 2000 and 2002, we were able to double the NIH, get funding of approximately 15% a year. Now, I just want to say, for all of you, I know you're interested in biomedical research, and you know the meaning in lengthening lives, making America competitive, and giving us great economic growth. I just want to say that this year is so disheartening. We all know there are tough budget constraints, but this is the first time in 26 years since I've been in Congress. I was in Congress in 1966. In 26 years ago, NIH has not received any increase at all. We are really flat funding. And we respect and understand the problems all your officers have. And we really want you to help us this year. We really want you to do something, whatever you can. You know, already on the Senate side, Senator Specter recognizes this problem. He is Senator Harkin put in an amendment to the budget resolution on the Senate side, adding $7 billion to the health education function, which would mean that the NIH, instead of being flat funded, although the budget resolution doesn't direct money to the NIH in that document, nevertheless it indicates that the NIH would get probably $1 billion. This is indispensable. Now this vote's coming up this week in the House, and we're not here in this caucus to exhort and implore and tell you how to vote or your offices, but we wanted to bring it to your attention. And we know that your members are genuinely interested in the NIH. Whatever you can do on that budget resolution in the House to adopt Congressman Mike Castle's amendment, which would add several $7 billion, including the health education and NIH budget, would be really greatly appreciated. And I know you understand the meaning of the result that we need to achieve. But today we're going to learn a lot more about gene therapy. I was just talking to the good doctor, and we heard a lot about gene therapy for a while, and then the issue died out for a while. There were a couple of bad results, as you know. Gene therapy continues to be touted as a form of molecular medicine capable of having a major impact on human health. Possible outcomes range from curing genetic effects to providing relief from diseases like Parkinson's, Alzheimer's, cancer, heart failure. The basic concept seems kind of simple. You get a gene and you inject it into someone along with trillions of cells that exist in somebody's liver or brain, and somehow you vector that gene, I hope the good doctor will tell us, into the person so some good result obtains. Or you can introduce rare stem cells into the bone marrow and ensure the production of blood for the rest of the person's life. Now we're privileged today to have with us a scientist who is known throughout the world, a non-scientist, Dr. Inder Verma. He's the chair now of the Laboratory of Genetics, the Salk Institute of La Jolla. What a pretty place to be from, Doctor. Dr. Verma received his Master's of Science in Biochemistry from Lucknow University in India back in 1966. He received his PhD in biochemistry from the Weizmann Institute of Science in Israel in 1971. He was a postdoctoral fellow for the Department of Biology at my own school, the Massachusetts Institute of Technology in Cambridge, Massachusetts, from 1971 to 1974. Dr. Verma.
Thank you. Thank you, Congressman. Thank you for the opportunity to come here and give a talk in the Congressional Building. It's a rare honor. You know, for us as scientists, we do experiments, results, but in fact there's a whole group of people here who actually make that possible for us to do it. And they are as big a part of our science as the scientists themselves. And science and society has to be forged together. And I always believed, and I still believe, that's the responsibility of the scientists to tell the society what they're doing. And I think if the society understands, which naturally the politicians is part of that, then they are likely to support more scientific endeavors in future. So I'll try to make it as simple, but at the same time, I'd like to give you some idea how we actually do the science. So as Congressman just told us, gene therapy is a form of molecular medicine which can have major impact on human health in this century. Now, for the layperson, the idea that you can actually cure a disease for which there exists no cure is really quite exciting. For scientists, the idea to study how a gene is expressed, where is it expressed, when is it expressed, how long the protein will be made is really scientifically exciting. But the idea that you can actually cure a disease for which there existed no cure is really quite revolutionary. Now, it was easy to understand when we talked about gene therapy for genetic diseases, and there are many. I just list a few. There are now hundreds now that we know the human genome map. Can you see it when I turn back? Okay. Let's take a case of a blood disorder. A child is born, 30,000 perfect genes, except one gene is deficient, broken down in a fashion that the child cannot now make its blood to clot. Whenever there's a cut, the blood does not clot because he is missing one gene, and its product is a protein that is responsible for clotting. And the idea is simple then, can we introduce that gene which will make that protein, and therefore this child will have perfectly normal life like every other child. And some other diseases I listed here. But I think the excitement of gene therapy is much broader because we can actually influence the outcome of what I call acquired diseases, cancer. 1 in 3 or 4 in this room will have a bout of cancer in their lifetime. And to date, it is still true that if one goes to the doctor, you get surgery, radiation, or chemotherapy, although new drugs are coming, but that's the usual modality. The idea that you can introduce a gene which can directly kill the cancer cell, the idea that you can boost the immune system so that it can recognize the cancer cell as a foreign, while hasn't come totally into fruition, I'll give you an example, but is really worth considering, worth revolutionary, because it is not invasive, and therefore it improves the quality of life. Similarly, neurological disorders, I'll give you an example. Can you influence the outcome of a disease like Parkinson or Alzheimer, which are going to be a major disaster for us as we live longer and have a better quality of life? So I'll venture to say, and I still venture to say what I've said 10 years ago, that gene therapy will have a major influence. The concept of gene therapy is disarmingly simple. Introduce the gene. Its product should either alleviate the defect, cure the disease, or slow down the progression of disease. The question which many will ask, and the title sort of reflects, why is it then there are not more successes in gene therapy if the concept is so simple? And I'll venture to say that it's not really the concept, it's the execution of the concept. How do you really introduce a gene in brain which has trillions of cells? How do you really introduce a gene in our liver which has billions of cells? How do you introduce gene in a hematopoietic stem cell which is probably 1 or 10 or 100 in a background of 10 million cells? So really the problem is, how do you introduce genes efficiently, safely, so that its product has the ability to do the job it is supposed to do? And there are many ways to do it. You can inject a needle with people, but all of them are not very efficient. The most efficient way that we know of are biological carriers, and those are viruses. One gets infected in the family one day with a flu, there's a good chance everybody gets it, and the whole body gets it very quickly because they know how to disseminate themselves. So we argued to use viruses as a tool. And here is the way we do them. Viruses have only one business in life. They just want to grow, grow, grow. They have no desire to kill the host, because if they kill the host, they kill themselves. So by and large, viruses have only one job in life, and that is to grow. But occasionally they acquire sequences, shown here in a sort of magenta color, which are causing the disease. So virus can be separated into two parts. A component which is needed to make more of itself, which we are interested, a component which causes the disease, and all what we are trying to do is to remove the disease-causing entity, substitute with a therapeutic gene, recreate a virus which is identical to what went in, except now instead of causing the disease, it is making the therapeutic protein. So in fact, all what we have done is to recreate a virus which has now the ability to do the product that we want it to make rather than cause the disease. So that's really the functional method how which we do these viral vectors. So what we like to do is what I call an ideal method of gene delivery. There is no perfect method, but at least on a computer slide I can at least have the fantasy of what I would like to have as a perfect vector. We like to have something we can make millions of it. And that's given, because if you want to introduce it in a brain or a liver, you need millions and millions of virus particles. And we can make 1 milliliter of virus particle which can contain 10 billion viruses in it. We like to introduce genes in dividing cell, like a cancer cell, which is rapidly dividing, so we like to introduce there. As well as in our brain, a neuron which is not dividing, we like to also introduce genes there. Ideally, we like to know where we introduce. After all, we have 30,000 genes, 3 billion nucleotides, 22 chromosomes. We want to introduce the gene where we want it to go in. It'll be nice to know. We can't do that, but it'll be a nice feature to be able to do that. It'll be nice to control the production of the foreign product, like a rheostat. Turn it on, off, when, how much, where. It'll be also very desirable that we can put a lot of genetic material. Majority of the genes that we're interested fall in the size of about 2,000 nucleotides to about 10,000. But there are bigger ones too, like muscular dystrophy. But we at least hope that we can entertain the notion of using majority of the genetic diseases to be cured by using this kind of a method. And of course, we want to have no unpleasant immunological consequences. Remember, our body is designed for billions of years of evolution, or intelligent design, whatever you prefer, to be able to fight the infections. We are very fortunate, right? I would never be able to travel in a plane if we didn't have an immunity system to fight these viruses all the time. And yet we are bringing billions of these viruses back to the body and now asking the body, oh, we are good guys, don't make any immune reaction. So we have to learn to do that also, and you will see that is a problem we have only partially solved. So with these kinds of things, what turned out was that what you need to have in science is some skill, patience, money, and lots of good luck. We were lucky. And the luck came in an entirely unexpected manner, and that came in the form of HIV. Now you will think I'm a nut to think luck comes in the form of HIV, but just give me a minute or so before you make up your mind. HIV is a very unusual virus. It has the ability to introduce genes in cells which are not dividing, like macrophages. People get dementia because it goes in the neurons. And yet it becomes a resident part of the body. So it has learned a lot of things which we like to have in a vector, a delivery system which can behave like HIV but not cause the disease. So that's really the goal we had, because it could do many of the things that we like to do in ideal vector. So we assembled a team at the Salk Institute. I don't show this team to you because they're all good-looking folks, but just to re-emphasize the following, that how American science to this date, certainly in biological sciences, so heavily depends on foreign students and foreign postdocs. Luigi Naldini is from Italy. Ulrika Blumer came from Germany. Didier Trono is from Switzerland. I'm from Bronx. No, not quite. And Rusty Gage is the only American hostage we have in this team. So it just tells you that the vibrancy of American science still is very much goes along with the generally having lots of good students come back. And fortunately, many of them are now able to go back and set up the labs in their own countries. So here was the problem. Here is the HIV. I have now made it in the sort of a linear diagram of all the genes that are involved in ability of a AIDS virus to cause the disease. We can divide it into two parts already. One part of the virus is making more of itself, which is what we like. One part of the virus here, all this, is causing the disease which we don't like. So what we like to do is to make an HIV virus where we eliminate all these genes, 7 of the 9 genes, and ask the question, can we still maintain its ability to introduce genes where we want and yet not cause the disease? So one by one and singly and multiply, we eliminated all these genes, And what we have now is the following, which thousands of people are now using worldwide, these vectors. We started out with something which had 9,000 nucleotides, meaning that's the full length HIV virus capable of causing the disease. We eliminated 90% of the genome. What we are now left with is only 9.9% or even less of that. And that is sufficient for us to be able to introduce the gene in any cell, where we want, how much we want, and when we want, without carrying the baggage of all these extra genes which has the ability to cause the disease. Now, of course, safety is always primary in these concerns. All I can tell you to date, hundreds of labs have used, we have not been able to recreate an infectious virus by using such a debilitated virus. That is not to say it might not happen one day, because we can only do so many experiments in the lab at a certain scale. I can't tell you if somebody starts making thousands and thousands of liters, the combination may occur. But to date, it hasn't, and the probability is not zero. There's never zero in biology. It's very low, because you need to introduce 9 genes before you can remake an infectious virus. So the question is how good it is and does it work? Let's start with the brain. Brain is truly non-dividing cell. There are some cells which are dividing, but most of them are neurons. So we argued what happens if we directly introduce the virus in the brain. Here we took 1 microliter of the virus, which contains about 30 million particles, meaning 30 million cells can be introduced this gene which can make the foreign protein. And shown here is, so the nice thing about brain is it's bipartite. On one side you can introduce the gene, other can be a control to know whether it functions or not. So we introduce here this side the gene, which when you shine light on it becomes green in color, just so that we can detect it. And here's a section from a mouse in this case. You see there's no expression on this side. There is a protein being made, and 90% of the cells where we introduce the gene, it's made, and it is there for the rest of the life of the mouse. Just one example. Another example, I give you muscle. Muscle makes 40% of our body weight. Some in NFL probably have 80%, but by and large, it depends how much muscle you want. We have about 40% of the body weight is muscle. Muscle are over these muscle fibers, and they'll be great factory to produce a foreign protein like insulin, factor 9, factor 8, which can then produce the protein, secrete in our serum, and go all over the body. So instead of injecting, wouldn't it be nice if the muscles could actually be a factory of making that protein internally? So here's a direct introduction of the gene again in the muscles. You can see these long green muscle fibers, green again because the gene that we introduced into it can shine light and see the color, and the controls don't show it to us. I give you one concrete example from eyes. Eye is a very useful tissue for us as scientists to study because it's external. You can easily manipulate. And there are hundreds of eye diseases, which some of them there is cure, and lots of them don't have no cure, and they're genetic diseases. One such disease is retinitis pigmentosa. In this case, you have advanced blindness in this case, and about 15% of the people who have this suffer from defect of one or the other specific gene. So we argued, can you introduce the gene directly into the eye? Now I give you just a little background of biology here. Eye, normally light comes in and hits, this is your retina. There are about 7 layers of retina. So there are 7 layers through which the light goes through here. Normally the light comes in, hits your retinal cells, and the message eventually goes to your brain. So light comes in here, message goes to the brain, and then you recognize, which is actually quite remarkable, because if you see Rembrandt's night watch, in less than a second you say this is Rembrandt's night watch, and yet the light has to go through your eyes, through all these colors, go to the back of your brain. The cortex has to make the message clear, and that all done in less than a second. That's another astounding thing of something we will have to one day understand. So we asked the question, can you introduce gene? Then there's a seat here if you like. There is a possibility, can we introduce these genes directly into the eye? So here is an example where we directly introduce genes in the eye, and again we hit the light on it, is green in color, and we can specifically introduce the gene in those cells where there's a deficiency. And I give you one example. So we created a mouse, and others created a mouse, where you deleted one gene from the genetic background of the mouse, the same gene which causes a disease called retinitis pigmentosa. As a result, these mice are blind because that gene is now gone, and as a result, they have none of these cells which are necessary for the vision. So here's the normal cell from that normal mouse. Here is the mouse from which the gene was deleted, and now basically they are blind because they have none of these cells which are necessary to get the light through them. So we argue now, since we deleted this gene from this mouse, which is now blind, if we put the gene back at birth, will this mouse be able to make these proteins which will allow it to become like a normal mouse, and will it restore its vision? So if we could do that, then perhaps it will be a good entree to go into the humans, where there is also partial blindness set in, and could you recover the same sets? Now remember, this is mouse from human, but that's the best experimental model we can use. So here's one such experiment where we took a mouse, injected directly into the eye, and here's the control, where a mouse where we did not put the gene in it, or something else we put in, but here put the gene in, and you can see 1, 2, 3, 4, or 5 new layers of the retinal cells are now formed, which are completely absent in the control. Now it's not perfect because these mice have to be manipulated very quickly, and they have to be done very, and I don't know if anybody have worked on ever mice here. A newly born mice is very hard to open its eye and put things into it. But nevertheless, this mouse showed us partial recovery of blindness. In fact, based on these results and additional results, now there's a phase 1 and phase 2 clinical trial precisely for this kind of a defect going from the mouse data. So we have a start of doing these experimental things using these vectors, which are now based on HIV. Of course, the desire of every gene therapy person is to be able to introduce gene in your stem cell. Now there are many kinds of stem cell. We shall not talk about embryonic stem cell today. We will only talk about adult stem cells. You and I make 10 billion blood cells every day. And the reason we can do that, because there's a master cell in the body called the stem cell, which has the ability to make every cell type in our body. Some of them go on to make all our myeloid cells, like the red blood cell, white blood cells. Some of them go on to make our immune cells, the B cells, the T cells, to fight the immunity. But they all come from a stem cell. So the idea is, wouldn't it be nice if we could introduce the gene in the stem cell, and every time the stem cell divides to make all other cell types, that gene will make the right amount of protein. So one cell will remain in our lifetime making billions of other cell types, and if we could find that cell to introduce the gene, it will produce the protein in every cell. That's really the dream of all gene therapy people. That's how we started out years and years ago. And the question is, it can do it. The trouble is the following. If I were to take 1 ml of your bone marrow, there will be 10 million, roughly, all kinds of blood cells, white cells, red cells, everything. The cell that we really want is only about 100 of them. And they don't look any different than the rest of them. So how do you find, I mean, not to use cliches, this is a truly needle in a haystack problem. How do you find those 10 or 100 cells in the background of 10 million cells, which one you really want? Because they don't look any different. But you can enrich them, and on the top of that, these cells have this very unusual property, they're not dividing. So it's very difficult to put genes in cells which are not dividing because they're sort of closed. The chromosome is closed, so you can't put the gene in it. So the HIV has this unusual property that it can introduce genes in cells which are not even dividing. And therefore, we thought this was an opportunity of lifetime to do this experiment. So we took then human cord blood cells, which you can easily get from doctors or many other places. We enriched them for what we call stem cells, not purified them, but enriched them. Then introduced them into the virus into them, which makes the protein, and put them back into the mouse, and now ask the question, does this mouse make the foreign protein? How long and how much? And this is really, to this date, the most exciting experiment for me, even though it was done now 5 years ago, 6 years ago, that 10 to 15% of our blood, so if you now take this mouse, where you took a stem cell, put the gene in it, now you take its blood out, 10 to 15% of its blood is making now the foreign protein for the rest of the life of this mouse. Now if you and I are making 10 billion cells, and even if 1 billion of them are making the foreign protein for the rest of our life, that is really the great victory for gene therapy. That means we are producing the protein forever. So what can we do with that? We can take the bone marrow, for example, introduce into insulin and put into a mouse which is diabetic, and now ask the question, do you make the protein? Here, for over 230 days when we stop the experiment, you're still making large amounts of the insulin protein. Or for that reason, you could use now liver. Liver is another tissue where we make lots of our protein. We can directly introduce the gene in the liver, and now in this case, for example, we have a protein which is making hemophilia, a clotting factor. We can introduce that, and that is now making for days of this protein also. So really, the methodology, at least in the small animal model system, exists, where we can use these viruses, which in a way you could call the worst thing that we can imagine, HIV. We have converted a sort of a foe into a friend, and yet it has acquired all the abilities that it can introduce genes, produce the protein for a sustained period of time, and now the question is how useful this will be when we go into the humans, and what else we have done with them before we go into the humans. And already trials have started, but let me give you some other examples. This is just to show you, although I won't tell you much, that you can also use them to directly introduce them to your tumor cells. So here is a mouse where a tumor was made by introducing a specific types of cells. Here's a direct injection of the virus which has the ability to kill those tumor cells, and you can see there's a substantial reduction of the tumors. Now, I don't put too much stock on these kind of experiments, merely to indicate that you have the formal ability at least to be able to do that. What precise tumor, what precise nature of the tumor still needs to be determined, but at least these vectors have the formal ability to do that. What I'd like to spend the next few minutes is to really one particular example is in the brain. What can we do using these kind of delivery systems in the brain? The brain is, I'm not the first to tell you, is a great tissue because it's one major thing is it's separated from the rest of the body. You can essentially introduce any gene directly in the brain without worrying about the rest of your systemic system. So you can actually, for example, and there is stereotactically maybe 30 to 50,000 operations done in the United States where you directly through a needle introduce whatever you want in the brain. For example, if you wanted to introduce gene directly into the substantia nigra, which is involved in Parkinson, can you make the enzyme which is deficient in Parkinson? Can you introduce the gene in the hippocampus, which is involved in memory and learning? Can you introduce the gene in the pituitary? So in other words, you know the regions of the brain. Stereotactically, you introduce whatever gene you want. And if you have 30, 40, 50, 60 million virus particles, each one making the protein you're interested, you can begin to see what influence it can have. So with that in mind, we began to ask the following question. We got interested in Alzheimer's, not because I'm getting old, but it's also something to take in future. It is really a most devastating disease. I think as we grow older, as the quality of life changes, will become a major, it has already become a major problem. A lot is known about it, but very little in terms of therapy that we have at the moment. So we argued in the one form of Alzheimer's, where you have Alzheimer plaques, neurofibrillary tangles, meaning you have these big plaques in the brain, they tangle, and they basically cause destruction of the neurons and eventually loss of your cognitive functions. So one day I was, I don't know, sitting in a plane or something, reading papers, and I noticed that there's an enzyme somebody had discovered which has the ability to be a protease, meaning if you have protein as a plaques, you can put this enzyme, it will dissolve the plaques. And we asked the question, if you can find a system of dissolving the plaques in the Alzheimer's, will it not begin to recover the function? Will that not be a way to do that? So we then made such a virus in which we put this protease and injected into a mouse which was bred to become an Alzheimer mouse. So these mice were created to be Alzheimer's. So what you do is you take the same protein which causes Alzheimer's in human, you introduce them in the genetic makeup of these mice, and 100% of them have the Alzheimer's disease by the age of 3 months, 6 months, 9 months, depending when you introduced it. So they're a great model system for us. So here, again, one side of the brain you can introduce the gene, and the other side you can then ask the question whether this has functioned or not. So here is one side of the brain. You can even see the needle coming in. It goes directly into the hippocampus, because hippocampus is the area, remember, which is involved in learning and memory. So here's direct introduction of the gene. And you can see now, here are the big plaques in the control, And after introducing the gene, after 12 months, you can see the size of the plaques are still there, but they're much smaller. More importantly, if you now ask the question, is there the destruction of neurons? If you now look at the sites where you just put an empty delivery system without putting the neprilysin, you see there are far less neurons. When you introduce the gene where you have introduced the protease, you have now much larger number of neurons, almost as many as in a control mouse which is not Alzheimer's. So this begins to give us some thinking and hope that we can actually begin to intercede with the progression of this disease by introducing a gene which has the ability, in this case, to dissolve the plaques, and is now the cognitive function recovered. Give you another example to show that the cognitive function. So the main happens, and this is one bit of biochemistry, the reason we get Alzheimer's is, at least one form of it, is there is called amyloid precursor protein, which gets clipped into smaller protein, and it is this magenta guy which causes all the problem. So many people argued that it will be nice if you could stop this enzyme which is necessary to cut this. If you'd stop this enzyme, this will not happen, this will not happen, and this will not happen. How do you do that? Well, the pharmaceutical company will make small molecules which can then be given as chemical. They can go into the brain and remove the function of this protein. Unfortunately, that hasn't happened because it's a big protein. That is not to say it won't happen one day, but it's still not done. So we wanted a proof of principle. So we argued, what if we block the function of this protein? Will we then prevent the formation of Alzheimer's in this mice? And more importantly, if we block it, it will not allow further formation of the plaques, and they should now recover their cognitive function like memory. I just want to show you that you can do this. You see these big plaques. By introducing this particular gene, you can block the plaques. And the more interesting question is now, what happens? So here is a control. You see the large number of plaques. When we block it, you see the plaques are much less, much smaller number. But more importantly now, ask the question, what happens to the function of the brain? What happens to the cognitive functions? So these mice are Alzheimer mice. They have lost their ability of memory, spatial memory. And the way you do this experiment is you have a very big tank of water. In the water, there's a small platform. So the mouse is put into the water, and the mouse runs around and eventually finds the platform. Otherwise, they will drown. The smart mice, if you put them many, many times, after a couple of trials, maybe 5, 10, 15 trials, they will go straight to the platform because they remember it now. The Alzheimer mouse just goes on and on. Takes it hours, sometimes minutes, sometimes, even though you train them, but they have no now spatial memory because they have lost that function due to, in this case, making plaques. Now, this is not a perfect assay system, but this is the best system we have. It allows you to now have spatial memory. So these mice have lost the spatial memory. They take much longer to find the platform. If we introduce the gene, we are asking, can we now reverse that? This is actually a very important experiment because what these mice already have Alzheimer's. We are asking the question, by putting this gene which dissolves the plaques, do we recover the function? So that's a great hope that you have Alzheimer, you can get some recovery of that. That's shown over here. So these are mice, 4 different kind of mice. Those which are controlled, that is to say they are normal, they should behave normal. Those which have Alzheimer, they should behave like an Alzheimer. Those which we introduce the gene which prevents them to become Alzheimer's, and those which we took a non-specific gene so it doesn't mean anything. Right? So there's a control. You always have to have a control so that the vector alone doesn't have an influence. And what you see here is actually better here, that the 3 mice, the normal mouse, the one where we put the gene which prevents the formation of plaques, or a control mouse, all recover function equally well, except the one where the gene was not put in, and that has lost its function. So these mice were Alzheimer's, but when we put the gene back, at least one part of their memory, the spatial memory, the cue memory, they now behave exactly like the others. Now, I'm not advertising that this is going to be a cure for humans in the long run because we have to cover larger areas of the brain, but that should give you an indication that you have the possibility of interceding. If you can now make large number of virus particles, if you can cover a larger area of the brain, you have a much better chance, at least giving you the hope that there is a possibility of recovering your cognitive function. Just to finish then, a couple of more things. You can begin to actually ask the question, can you create also models by this kind of methods? It is now well known through epidemiological studies that people who have a certain form of a gene called ApoE4, this is a gene involved in our lipid metabolism. People who have one kind of a gene, they have multiple different kinds of genes called ApoE4, you can see these people will have an early onset of Alzheimer. In fact, 50% of them will have Alzheimer by the time they're 50 or 60 years old. As compared to majority of us which have this form of protein called ApoE2, they will all have Alzheimer's too, but much later in their life. So the question is, this is just an epidemiological study. Is it true? Can we convert into the mouse? So we took this mouse, introduced into it ApoE4. So remove the ApoE2, put the ApoE4, and it's really remarkable. You can see much greater number of plaques now when you put ApoE4 as the controls. So we can actually use these methods not just for gene therapy, but you can now use it for a wider context of biology to begin to make model systems, and we're doing the same thing in the monkeys now because they're closer to human in terms of doing many Alzheimer's studies. Finally, it's really not useful to just keep putting the gene. You like to control it. How much? Because you don't want to put a gene that you can't control. You like to be able to control it. So here's a vector which we made, which you put in the brain. You see some amount of protein being made. Again, shining light on it becomes green in color. Now you may give it a small molecule from the water, and now you see there's a tremendous induction of that protein. In fact, you can control it. Turn the gene on, off, by just giving a small molecule in the water. If they drink it, a few days later the gene is turned on. You don't have it, the gene is shut off. Again, these are not perfect systems, but that's the direction gene therapy is going, that we don't think it's useful in the long run to always just have the gene, but we like to regulate it also. And this gives you an example that this is possible. We can also make, I'll take 2 more minutes. Do I have still 2 more minutes? Yep.
Absolutely.
Okay. So we can also begin to use these kind of things to make models for cancer models. One of the biggest diseases, we have many cancer diseases which are lethal, pancreatic diseases, glioblastoma is one of the few diseases which is extremely lethal, and we don't even understand where it originates. So we are interested to study where does the gene, what kind of an area in the brain, what type of a cell in the brain is really making glioblastomas, because if we don't understand that, we will never cure it. So this is just to show you it's a terrible disease. The lifespan once you have glioblastoma is really very short. So we now know, we want to know what part of the brain the disease starts. We don't really know them because the only time we see the disease once it's already there. And unless we know what type of a cell it is, we will not be able to intercede with the disease, at least in a very specific fashion. So there are 3 or 4 regions of the brain where we know neurogenesis occurs, means cells grow. This is called these are the 3 regions where, where we can introduce the gene. In this case, the genes we know. So once people have tumors, we then cut them up and we know these genes have gone bad in these people. So we know the genes which are involved in causation, but what we don't know what cell type it is. So we take these genes, the bad genes which cause the disease, put in this lentiviral vector that I showed you, directly introduce into the brain in a place where we want to know what kind of cell that's making it. And now we were amazed to see, you can almost see this mouse actually. It has a big head here. Within 60 days, 90% of all the animals we injected had this. I'm sorry, this is lunchtime, but just one bad slide here. These are massive glioblastomas, exactly the same type as the human. So I'm only adding this to you that when we talk about gene therapy, you're only talking about a disease, but in fact you can begin to use these kind of methods for much broader understanding of biology, and here creating a model system how you cause the disease. And this is, I think, the first opportunity we'll have to deal with in terms of precise cell type. So finally, let me finish by telling you a very brief history of what happened to gene therapy. Gene therapy really came into existence in the early 80s, and the concept of gene therapy was really 81, 82, and my first paper when I was involved was in 83. That was one of the earliest papers. In about mid-80s, we began to understand how to put genes in bone marrow, but not very well. We began to understand, so we began to sort of play around with animal model system. Now remember this, 25 years ago, hindsight is 20/20. We should not have done many of the experiments, but we didn't know any better. In fact, in 1990, French Anderson at NIH, along with his colleague with Mike Blaise, did the first human clinical trial where they took 2 patients with ADA. ADA is the deficiency which the bubble boy, David the bubble boy, had, and asked the question if they introduced the gene in their T cells. So they took their blood cells, introduced the gene, and there were really great hopes. Everybody was excited at that time, but it turned out it didn't work. It didn't work because now we know why. It didn't work because we learned that the genes which were introduced were shut off very quickly, and nobody had realized that before. Only when they did the experiment they found out. At the same time, number of other disappointments occurred. There was an LDL trial, there was the cystic fiber. So all these suddenly begin to realize that we are moving much too fast in the clinic without understanding the basic biology. So Harold Varmus then, who was then the NIH director, said, slow down, created a committee to look into it, how we can improve the quality of science so that we do more basic science. And that happened. But then unfortunately, many of you might recall, in mid-90s we have death of Jesse Gelsinger, who got adenoviruses for a gene, in this particular case, for a short period of time, but Jesse Gelsinger died. And that really, in fact, I was here when Senator Frist was having the hearing to give I was the president of American Society of Gene Therapy to give the testimony to say, yes, we didn't really understand much about it, and therefore perhaps in retrospect we may not have started this trial. But nevertheless, that left a big pall on gene therapy, considerable consternation among people whether gene therapy, in fact the headline said, gene therapy kills but not saves. So it's not pleasant to be in a field when you read these things. Nevertheless, it allowed us to refocus, spend more energy on understanding basic science. And I finish by my last example here is that now we have success in France about children who have gene therapy given to them because they have the same sort of a David's bubble boy disease. 11 of the 14 are fine, but 3 of them got cancer. So again, suddenly there came the concern, what happened? And I finish it by showing you one interesting. So the reason we think it happened, cancer, is because the virus went into a cell, into the chromosome, went next to a gene which causes cancer. That's the explanation. So we said, is that really true? So we took the same gene, which the French group has, and this paper should come out shortly in Nature, maybe tomorrow or day after tomorrow. And what happened is that we now found that the gene they used itself, not the patient, just the gene itself, 33% of the animals, so this is survival, 100% of animals survive, but when we put this gene, which is supposed to be therapeutic, this itself causes 30% of the animals to die of the cancer, the same cancer as those children had. Now, this doesn't mean the French didn't do the right trial. It only means we don't understand these things, and the only way we'll do it, they went to the clinic, now we are back from the clinic to the bench, and from bench it has to go back to the clinic. I think that's the only reiterative process by which success in science will come, is a constant back and forth. We need to know from the clinician what happened, then we can go back to the bench. So, there are 765 current clinical trials worldwide. There are at least 10 of them in phase 3. Majority are still in cancer because, again, the diseases are very lethal. So the field is not dead. The field is not, as contrary people think, it is vibrant. It doesn't have the support of big support at the moment from industry because they don't quite really know how to deal with cell therapy in general. And we are hoping that NIH will continue to support this because NIH has been the large funder of the gene therapy in the past. And finally, this is my status in gene therapy. I started out as a young man. I will never be allowed in a plane now, by the way. I'll be always checked out. And my students told me, how long will it take before gene therapy will work? And this is what they think. This is how old I will look. Finally, it is not possible for anyone to do this work alone, certainly not me. I have an enormous number of colleagues over the years. Some of them are listed, and the work is done at the beautiful Salk Institute, which is on the right coast, on the other side of the country. Thank you very much for your attention. I'll be happy to answer questions. So, definitely 18 children who are suffering from a disease called a gamma C deficiency, meaning, let's just put in a generic term, they have no immune system, like the David bubble boy. they would almost surely be dead, because they can make T cells, they can make B cells, and in fact, unless you give them a bone marrow transplantation, which is done, but bone marrow transplantations are hard when you don't have a matched bone marrow, but can be done. Of those 18 now, 15 of them, and 13 of them are now over 4.5, 5 years actually, they are like normal kids. They get infections, they have flu, they have tetanus shots to given to them, they make B cells. Now, maybe they might get tumors because the virus is integrated. It is possible one day. We don't know that. But these kids are living normal life.
Are they still in that bubble that I saw once?
No. They are normal life. Right. See, and that is, if you think about it, if it was my kid, my God, that is amazing. Now, 3 other children in Italy have another disease called ADA. There are several of them in the late clinical stages of cancer therapy. One of them is a company, and I should say up front, I was one of the founders and now on the board of it, called Cell Genesis. It's a company which is involved in prostate cancers, where they take a virus to introduce a gene called GM-CSF, and they are in phase 3 clinical trials. In phase 2, they were able to show the extension of the life from 22 months to the median age is now up to 30 months. So there are clearly examples. There are very few at the moment, but just these 2 or 3 examples tell you, yes, the system itself, the concept itself can work. And certainly for these kids, it's working very well.
Let me ask this. Your ideal situation, doing the research you're doing, and you said you had NIH grants helping you. What would be an ideal? Tell us in the next several or decade of years. What could you do first, do you think, if it were an ideal situation?
Well, certainly for us, as I said, my career, I was 26 years old when I started my first grant, was an NIH grant, $150,000. It was an enormous, without this I would never have been able to do anything. So this is a marvelous country to give an opportunity to somebody that young to be able to do things. And NIH has been a great support, but of late, I don't want to sound like a crybaby here, we have a hard time. I have written 9 grants since January, and I'm hoping I will get 1 or 2. It's not that I'm doing bad science. It's not that I'm the great science. When you're funding only 9 or 10% of the 100 grants that come to a study section, I think you can close your eyes and throw darts. You can probably get the 9 best ones. So I think the sad part is this is the time when science is blooming. The genome map is there, the genetics is there, the chip technology is there, and actually small molecules are coming up to affect the cancer. This is the time not to retreat. And I think what happens in science is that if you don't have a source of sustained funding, it slows down. And then to bring the momentum again is to bring the same guys who are well trained is very hard. So again, I'm very grateful, very thankful for all the governmental support. But I think this is the time to keep it going, because I think, in my own mind, when the history of the world is written, the National Institute of Health will be the beacon which the whole world will look at.
You went unexpected. Now, I'm going to ask you again. What's the ideal situation in 10 years from now? What would you like to see as an ideal happen with your research?
Well, for my research, I would like to see, first of all, more young people taking this, being trained in our lab, who has the ability then to go further, use different diseases, use different modified vectors, use more safe vectors, use better designed vectors, and actually have a much more targeted therapy that you introduce the virus, it only goes where you want it to go. That's what I like to see.
So, Inder, you're convinced that at some point down the line, it's going to work. You're going to introduce genes and cure diseases in people?
I have no doubt it is already doing it. If I wasn't convinced, I wouldn't come here. What I regret the fact is that we should have perhaps spent more time understanding how these viruses work. I mean, the viruses have billions of years of advantage, and we are given 5-year grant cycle to prove that point. That's tough. That's tough. I'm not asking billion years.
Yes, ma'am.
Yeah, I noticed that 9 of the current trials are non-therapeutic. I assume those are outside the United States, and I was wondering where they are and what they're trying to do.
So lot of the trials are actually, some of them are in Germany, some of them are in France, in England, and a lot of them has to do with the AIDS virus. A lot of the trials are for HIV.
And that's not therapy?
why it depends what you're doing it, if you're putting it to block it or if they want to accentuate it. So I don't really know the details of those trials because I'm not involved in them, but that's what I suspect they are.
Tim?
Thank you. That was an excellent talk. I'm at the Institute, and I think it might be helpful to hear a little bit more about the RAC and how that's trying to address some of the issues in terms of the concerns of safety.
So RAC is an organization which is now, I think, called Office of Bio OBA. I think that's the new designation. So RAC, I was a member of the RAC for years, so I know a little bit about it. RAC and FDA, so there was always a question. RAC has no regulatory authority. By and large, it was more an advisory body. So people brought their protocols to the RAC, and the RAC then looked at it, listened to them, and asked for the questions whether it was safe or not. And then it went to, people also had to go to FDA independently. And FDA has, of course, as you know, statutory regulatory, they could say yes or no. So for a while, it wasn't clear that FDA had already approved, then they came to RAC, it didn't make much sense. But RAC, on the other hand, has a big punch, because if an institution which is working on a gene therapy and doesn't have RAC approval and they have NIH funding, they can stop them. However, if you were not involved in NIH funding, and RAC was meaningless because you could do what you wanted. But now I think there's much more harmonization. So I believe RAC has served an enormously important purpose for not only safety, but also actually I think improving the quality of science, because they were a bit of a censor to say, this doesn't make sense. And they were able to do that. So I think, my opinion, RAC is working an extremely valuable function.
Yes, sir.
Yeah, thanks. It actually gets somewhat to my question. Which was whether there are particular regulatory barriers or ways that we're not prepared to adequately, regulate and evaluate this sort of technology kind of going down the road. And the second part is whether there are other nations that are better equipped to effectively and efficiently give regulatory approval to these sorts of technologies.
So let me answer the second part. Almost everyone in the developed world or even developing world, like India, where I come from, looks toward FDA as the beacon. They basically use their guidelines. So I don't think we learn so much. Everybody learns from us. Now, whether the process is moving far, again, I'm not a clinician, so I can't really answer precisely if I have ever suffered or an industrialist or from that point of view. But from the regulatory point of view, I think my impression has always been RAC and FDA has bent backwards to be helpful and supportive to be people. Now, maybe in certain cases they've asked questions but not pertinent, I don't know those. But my impression, and I've spent many times on many meetings because they arrange special meetings, they're always trying to find out what you need to do and what you need to do to explain, to do experiments to prove that. So again, from my perspective, they are not a hurdle. Yeah. I don't think so. That's not my impression.
Any further questions? Well, Dr. Verma, thank you very much for an enlightening talk.
Thank you.
Excellent. Okay. Good to see you.
Thank you.